Your doctor lives by a code. First, do no harm. That sounds like something from a bygone age. Because truth be told, there's a lot of harm done by doctors. This channel will tell you exactly how. Let's begin the 50% promise. If I tell you a medicine reduces your risk by 50%, your brain has already reached for its coat. We're leaving with the prescription. That's because 50% is one of those numbers that arrives wearing a cape. It sounds decisive, scientific, and reassuringly expensive. Now for the detail nobody puts in the headline. Suppose your chance of developing a disease over the next decade is 2 in 10,000. Take the medicine, and your chance falls to 1 in 10,000. Congratulations, you have just experienced a 50% reduction in relative risk. You have also experienced an absolute risk reduction of 1 person in 10,000. The medicine didn't remove half the danger from your life. It removed 1 chance in 10,000. Neither statement is false. They are simply engineered to produce very different emotional reactions. Relative risk tells you how much smaller the risk became, compared with where you started. Absolute risk tells you how much your actual odds changed. One is a ratio, the other is reality. This distinction matters because human beings are spectacularly vulnerable to large percentages detached from small numbers. We hear risk halved and picture disaster narrowly avoided. We rarely stop to ask whether the disaster was likely to occur in the first place. Imagine a lottery ticket advertised as offering a 100% greater chance of winning. Technically true if your odds improve from 1 in 20 million to 2 in 20 million. Still not worth planning where you'll park the yacht. Medicines deserve the same skepticism. A treatment that halves a tiny risk may be worthwhile. It may not. That depends on side effects, cost, inconvenience, your personal values, and whether the disease itself is catastrophic or merely unpleasant. Those are judgment calls. But they should be informed by numbers that describe the world as it is, not merely as it sounds. Whenever you hear that a medicine reduces your risk by 30%, 50%, or even 80%, ask one question before allowing yourself to be impressed. 30, 50, or 80% of what? That single question separates statistical literacy from statistical theater. Now there's a new kid on the block, medically speaking, and the hype around the drug is blasting its success. But you're a research fundee, and you just have to get your facts in formation before you try anything new. So you search the medical literature and find study after study reporting positive results. The evidence appears overwhelming. Scientists agree. Journals agree. The data, apparently, agree. Now imagine discovering that the studies you found are only the ones that made it into print. Welcome to what researchers politely call publication bias. Everyone else calls it the file drawer problem. The name is wonderfully mundane. It conjures images of dusty cabinets and forgotten paperwork. Unfortunately, that's not far from reality. Suppose 10 independent clinical trials are conducted on the same drug. 3 show a meaningful benefit. 7 find little or no difference between the drug and a placebo. If all 10 studies are published, readers conclude the evidence is mixed. The drug might work, but the case is hardly overwhelming. Now suppose only the 3 positive studies appear in journals, while the 7 disappointing ones remain in filing cabinets, hard drives, or abandoned grant reports. Nothing about the drug has changed. Only your view of it has. From the outside, it now appears that every published study supports the treatment. A doctor reviewing the literature sees unanimous agreement. A journalist reports a breakthrough. Patients wonder why anyone would hesitate. The medicine has acquired a reputation that the complete evidence never earned. Why do the missing studies disappear? Not because researchers necessarily set out to deceive. Scientific journals prefer exciting discoveries to papers whose conclusion is essentially, "We looked carefully, and nothing much happened." Researchers build careers by publishing novel findings, not by confirming disappointment. Funding bodies like success stories. Pharmaceutical companies have little commercial incentive to spend time and money promoting results that fail to promote their products. Even editors and reviewers are human. Positive findings simply make for better copy. Each decision is understandable in isolation. Collectively, they create an optical illusion. Imagine judging a cricket batsman after watching only the boundaries, or evaluating a restaurant after reading only the 5-star reviews. You would not be lying to yourself. You would simply be making a decision with half the evidence missing. That is publication bias. The danger is not that published studies are false. The danger is that published studies may not be the whole story. A medicine should be judged by all the trials that were conducted, not merely by those that escaped the file drawer. Because when evidence disappears, certainty has a curious habit of appearing in its place. Clinical trials are another dodge at the truth, the whole truth and nothing but the truth, because the sample group is often assembled on a very curious basis. If you've ever watched a cooking show, you've probably noticed something curious. The chef always has exactly the right ingredients, the right equipment, and apparently unlimited counter space. Nothing is burnt, nothing sticks, nobody's toddler wanders in and licks the butter. Real kitchens bear only a passing resemblance. Clinical trials work in much the same way. Before a new medicine reaches your doctor, it has usually been tested on a carefully selected group of volunteers. They are, statistically speaking, the easiest people in whom to answer one question: does this drug work? To answer that question cleanly, researchers remove as many complications as possible. The volunteers may be between 30 and 50 years old. They may have just one medical condition: no kidney disease, no liver disease, no diabetes, no autoimmune disorders, no pregnancy, no 6 other prescriptions rattling around in the medicine cabinet, no unexpected variables if they can be avoided. From a scientific perspective, this makes perfect sense. If you're trying to determine whether a drug lowers blood pressure, you don't want dozens of unrelated factors muddying the picture. Every additional illness, every extra medication, and every genetic difference introduces another variable that makes the results harder to interpret. It's rather like trying to judge the quality of a violin while someone is operating an electric saw next door. Researchers aren't necessarily trying to make reality look prettier. They're trying to make it quieter. The problem is, what happens next? Once the drug is approved, it leaves the laboratory and encounters people, real people, the sort of people who forgot where they left their reading glasses because they're already wearing them. People who have diabetes, arthritis, and high cholesterol. People taking 4 prescriptions before breakfast. People whose kidneys have worked loyally for 78 years and are understandably becoming less enthusiastic. In other words, exactly the people who were often underrepresented or entirely absent in the original trials. Imagine a new blood pressure medicine. The trial includes healthy adults aged 30 to 50, all with uncomplicated hypertension and taking no other medications. The results are excellent. Blood pressure falls. Side effects are minimal. Everyone congratulates one another. Now the medicine enters everyday practice. The average patient is 68. She has diabetes, mild kidney disease, high cholesterol, a thyroid condition, and a collection of tablets that requires its own weekly organizer. Suddenly, the picture changes. Perhaps the medicine interacts with another drug. Perhaps reduced kidney function alters the dose the body can safely handle. Perhaps it still works, but not nearly as well as it did in the pristine conditions of the clinical trial. None of this means the original trial was fraudulent. It means the trial answered a narrower question than many people assumed. Not, does this medicine work for everyone? But, does this medicine work under these carefully controlled circumstances? Those are not the same question. This is why doctors continue learning about medicines long after they are approved. Many important side effects aren't discovered until millions of people begin taking a drug in circumstances that no clinical trial could fully reproduce. Medicine doesn't suddenly become less scientific once a drug reaches the pharmacy. In many respects, that's when the largest experiment finally begins. The lesson isn't to distrust clinical trials. Without them, we'd know far less than we do. The lesson is simply this: whenever you read that a medicine has been proven to work, the next question shouldn't be whether it worked. It should be, for whom? Because in medicine, as in life, the average patient has an irritating habit of not existing. It's the old saying, buyer beware. Whether it's eggs from the supermarket or drugs from the pharmacy, always make an informed choice.